Hello learners and welcome to Chapter 11 Part 2. Our focus is going to be on graded potentials and action potentials, but I also want to start us off with a synapse. Now, you'll see that this is a slide that I just included, and it's basically going to be focused on focus figure 11.3 in your text. So just make sure you refer to that focus figure to help you get through. Now, before we even talk about a chemical synapse, we have to talk about more membrane proteins. We understand the leak proteins that are always open, like the sodium and potassium leak protein, or leak channel proteins that are always open, allowing potassium to go out and sodium to come in. So we know that, and we know that potassium leaving is going to help us create that resting membrane potential. And we also know about the sodium-potassium pump. And what do we know about that? We know that the sodium-potassium pump is going to maintain that electrochemical gradient, maintains our negative 70 millivolts. Okay. Now we have two more different types of membrane proteins we're going to introduce. That's going to be ligand gated and voltage gated. On ligand gated, can also be considered ion gated. Okay. So just keep that in mind. I normally say ligand gated, ion gated. It also can be receptor. Okay, receptor channel, anything you want to call it at that point. Consider ligands like a chemical key, okay? And what's going to happen is that ligand or chemical key is going to bind to a channel receptor or bind to a channel's receptor. So remember, we can have proteins here that have certain channels. And then once that ligand binds to the channel, it allows it to open or close. And it'll allow it to change shape. So let me go ahead and see. Let's say it'll change shape to look like this. Okay. And when it changes shape, that's where we can have other things like specific ions cross the membrane. Let's say we have a sodium that's going to cross the membrane now because of that ligand gated channel. So that's the reason why you see it be called ion gate, ligand gate, or even receptor, because there has to be a receptor that's on the actual membrane protein in order for that ligand to be able to bind it. Little details like that, okay? Now, voltage gated is going to be meaning that it's a gate that's open with a voltage change. So we're talking about graded and action potentials. It's going to be important to understand voltage gated membrane proteins, specifically when it comes to action potentials, okay? Why? Because that's going to be a huge change in the membrane potential through depolarization, repolarization, hyperpolarization, and of course, getting back to rest. Now, again, that means there has to be some type of electrical trigger, and we usually focus on that with action potentials. Ligand gated, we usually focus with graded potentials, and we'll talk about that as we go along, okay? Now, I want to talk about a synapse, because in order for a graded potential to exist, and of course, we need the graded potential to be able to hit that trigger zone or initial segment or the axon of helic so that we can reach that negative 55 millivolt threshold and trigger an action potential, right? So you have to make sure you understand what a chemical synapse is, because a chemical synapse is going to provide that stimulus that gives us graded potentials. So here, let's orient ourselves. We have a neuron. As you can see, we have our soma, cell body, and we have our dendrites. And we also have our axon and the axon terminus. Okay? So we can kind of break up our neuron that's receiving the signal to the one that's sending the signal. If you are sending the signal, you're talking about axon terminus. So that's the end of an axon, as you can see here. And what we're doing is illuminating that piece of the axon terminal and the actual membrane of the dendrites and, of course, the neuron to be able to focus it into a larger photo. Okay? So presynaptic is what we call it when you are sending a signal. Postsynaptic, post meaning after, is when you are receiving a signal. Here, you see this is a presynaptic neuron. This is the axon terminal here from the axon. And then we have a postsynaptic neuron, which is going to be our dendrite and, of course, our cell body that's going to be receiving the ability to create a graded potential. Okay, that's the whole goal. Now, we're going to talk about graded potentials and action potentials as we go along. First, we'll get into graded potentials, but in order for the synapse to happen, we do have our axon here, and an action potential has to reach down to these axon terminals. So you'll see we're going to have that electrical signal go down the axon, and then it'll release down into the axon terminal. So what happens? The action potential is going to arrive at the axon terminal. When that happens, it's going to open a voltage-gated channel. Now, voltage-gated channels that we normally see will be for sodium and potassium, but we'll also see voltage-gated channels for calcium. That's Ca2+. So the action potential is going to arrive here. You're going to open up calcium voltage-gated channels, and that means calcium is going to rush into the actual cell, right? Or you can also say it's going to rush into the presynaptic neuron. Once it's inside the presynaptic neuron, then what's going to happen is it's going to cause or create synaptic vesicles, okay? So again, we're going to see these vesicles, and inside these vesicles are going to be neurotransmitters. Like the name suggests, neurotransmitter, it's a little tiny chemical messenger that is going to transmit a signal. Where is it going to be transmitting a signal to? The synaptic cleft. So we weren't able to see it in this visual, but in this visual, we see that there's a space, a tiny little space in between the presynaptic neuron or the axon terminal and the postsynaptic neuron or the cell body and dendrite. And that space in between is called the synaptic cleft. And so that neurotransmitter is going to, or again, that little chemical messenger that's going to transmit a signal, and these vesicles are going to go to the end of that axon terminal membrane, and then through exocytosis, they're going to release. Now, exocytosis means exo, the cell. Cyte is cell, exo means exit, so it's going to exit the cell. It's going to fuse with that plasma membrane and allow those neurotransmitters to be released. And guess what these little neurotransmitters are, folks? Yes, they're chemical messengers, but they're also our ligands. So what's going to happen is we have these different ligand-gated channels here, and the neurotransmitter is going to bind to a ligand-gated channel on the postsynaptic neuron, okay? Once it actually binds to that particular ligand-gated channel, it's going to open other ion channels or allow other ions to now come through, okay? When that happens, this is where we call a stimulus, right? Remember we were talking about graded potentials, how some can be strong and some can be weak? Well, the stimulus is going to be given by those neurotransmitters going across the synaptic cleft to these ligand-gated channels. And the more of that stimulus we have coming in, the stronger that signal. So it's going to be at this synaptic cleft, right when the neurotransmitter is basically hitting those ligand-gated channels, where you're going to have the strongest signal from the stimulus. This is going to trigger like sodium coming into the cell. So that means you're going to have a rush of positive ions coming into the cell. If you have a rush of positive ions, and we know our resting cell membrane potential is negative 70 millivolts, that's going to make the inside of the cell in this area more positive or depolarized, right? We have those little details. It can also have different ions that are coming in that are negative, like chlorine. Chlorine comes in, is going to make it go from, instead of being depolarized, it's going to be hyperpolarized. It's going to go below the negative 70 millivolts, okay? So just little details like that you can remember. Now, after the neurotransmitter has done its job, then guess what? There's going to be certain enzymes, yes, proteins, other proteins that are coming around to usually break down or basically disconnect the neurotransmitter from the actual receptor site on the membrane transport protein, okay? And when that happens, then the neurotransmitter is no longer opening that particular ligand gate. It will be closed, and either it will be reused because it's an enzyme, or it can also just be broken down with other enzymes. All right, folks, I want to get this in our heads, because as we go along, we talk about graded potentials and also action potentials. It's going to be important because it's going to be like, oh, here's a ligand-gated channel that's open. Here's a voltage-gated channel that's open. And we have to kind of understand the distinguishment between those two. Okay? Leak channels and also our sodium-potassium pump, we already kind of have seen before. All right, let's go ahead and get into graded potentials.